Craniopharyngiomas are the most common intracranial tumor of non-glial origin in children, comprising up to 10% of pediatric brain tumors. It's formed from the ectodermal remnants of Rathke's pouch and are composed of squamous epithelium. This pouch arises from the roof of the mouth of the embryo to become the anterior lobe or the adenohypophysis and ascends upward to meet the another part of the pituitary, which is the diencephalic diverticulum that will be the posterior lobe or the neurohypophysis. So the remnants of the Rathke's pouch are found mainly in the anterior lobe of the gland and the anterior part of the stalk. Craniopharyngiomas can occur anywhere from the floor of the third ventricle to the pharyngeal tonsils, with 67% being found in the suprasellar region. Although they are histologically benign, they behave aggressively, invading adjacent structures, making resection difficult. We know that this tumor usually contains cysts, so we have an important question. What are the ectodermally derived cystic epithelial lesions in the brain? Alright, it's a very important note. These lesions are arachnoid cysts, Rathke's cleft cyst, epidermoids, and dermoid cysts. All of these lesions typically exert their mass effect on the chiasm, the pituitary, the ventricles, and the hypothalamus, leading to visual disturbances, headaches, pituitary, and the hypothalamic dysfunction. But today, we will focus a little bit on craniopharyngioma. It has two different phenotypes corresponding to two different genotypes. Number one is the adamantinomatous craniopharyngioma, which is the classic and the more common type and usually occurs in children. Number two is the papillary type, which is less common and usually occurs in adults. Regarding the clinical picture, they are similar with very little differences. The common presentation of both of them include visual disturbances, which is the most common and the earliest presentation. Also cognitive impairment, personality changes, and obstructive hydrocephalus occur. But regarding the endocrinal disturbances, there is a little difference. The adamantinomatous type is associated with growth hormone deficiency in most cases, about 75%, while the papillary type is usually associated with hyperprolactinemia. Diabetes insipidus usually occurs in the adamantinomatous type. So, let's know what's the meaning of these two pathological terms, adamantinomatous and papillary. Adamantinomatous word is derived from the Latin word adamantine, which means very hard or stone. Now you can expect that this type undergoes some degrees of calcification. You are true. Let's take a look at this axial CT image. We see a mass located around the cellular region with circular pattern of calcification. This is the typical appearance of adamantinomatous craniopharyngioma in CT. The genetic mutation here is in the exon 3 of the CTNNB1 gene that encodes beta-catenin protein. This protein ensures the integrity of cell-to-cell junction. This type is more common in children. As regards papillary phenotype, when you hear the word papillary, you will expect that there is a capillary network over a fibrovascular support, and this is right. This type of craniopharyngioma is less common and occurs in adult age group. The mutation here is different. It is located in the BRAF gene in the amino acid number 600 in its sequence. Here, valine is substituted by glutamate. As we remember, glutamate is excitatory to everything in the CNS, even in the cell cycle. This substitution makes the division of the cells uncontrollable. So this mutation is called V600E mutation, while V means valine. 600 is the number of the amino acid, and E means glutamate because it is excitatory. So this mnemonic helps us to remember, as calcification word starts with the letter C, The genetic mutation here is in the CTNNB1 gene, which also starts with the letter C. Also, the papillary word, which starts with the letter B, is associated with the BRAF gene. Uh, uh, and this BRAF word starts with the letter B, with the differences between the two B letters kept in our mind. As regard radiological appearance, here we see a coronal and sagittal T1 weighted images, both of them after contrast and demonstrating a very heterogeneous mass with solid and cystic areas and variable enhancement, expanding the cella and extending into the suprasellar cistern and the interpeduncular cistern, causing compression of the third ventricle and resulting in hydrocephalus. All of these findings are characteristics of craniopharyngioma. Of course, there are different locations for craniopharyngioma, as it can be confined to the cella, can be pre-infundibular, retroinfundibular, purely infundibular, or extending up to the third ventricle, or even confined to the third ventricle. And so there will be a different surgical corridor and a plan tailored for every case. The papillary type usually appears as a solid mass, and if it forms a cystic lesion, the content of this cyst will be clear, not machine oil-like as in adamantinomatous type. As regard histological appearance, this slide typically shows the characteristics of craniopharyngioma. The squamous cells are packed together to form what is called the stellate reticulum, which is labeled by the yellow arrow. There is also peripheral palisading, labeled by the blue arrow. There are also rounded structures, labeled by the thin black arrows, and they are very characteristic to this tumor. They are called wet keratin particles, formed by the fusion between many tumor cells which lose their nuclei and become ghost-like cells. This keratin frequently calcifies, and then we will see these foci of calcification. Also, we see another finding, which seems to be a cleft. This cleft is called cholesterol cleft. We can see these clefts clearly in this slide. As these cells are full of cholesterol, many of them swell and rupture, pouring their cholesterol content, and this content appears clear because it is fat, giving the appearance of a space or a cleft. Now let's know some important surgical details about craniopharyngioma. Contrary to pituitary adenomas, the strong connective tissue capsule of craniopharyngioma adheres to the surrounding tissue, i.e., the optic nerves, optic chiasm, carotid arteries, infundibular stroke, and hypothalamus. This close attachment of the tumor capsule prevents the surgeon from the total removal of the tumor, because the traction over this capsule from hypothalamus, for example, will tear the perforators of the hypothalamus. Nothing is gained by leaving the patient with an infarcted hypothalamus after what we call a successful total removal of the tumor. We therefore do not attempt to remove the tumor capsule over the posterior superior aspect, i.e., from the hypothalamus. Also, any part which adheres closely to the optic nerves, optic chiasm, or arteries, we deal with it in the same way. We leave it in place. The objective of operation is reduction of the tumor mass, decompression of the surrounding structures, and the patency of the ventricular pathways. The needle is first introduced into the anterior aspect of the presenting tumor. This part of the tumor is invariably cystic, so we can evacuate the tumor and decrease its size before the section. Care is taken not to spill out the cholesterol-containing fluid, because it can lead to chemical leptomeningitis. Thanks for watching.